Hello, welcome students. We are studying the cell, and in doing this study of cell, we have to study the cell wall. We have to study the cell wall. This is the cell wall, and found in plant cell. You know that cell wall is found in plant cell. It surrounds the plasma membrane. It is found, or it is the cell wall, and so it is found around the plasma membrane. About the plasma we have studied in the last video. So it surrounds the plasma, and cell wall is non-living. Cell wall is non-living. It protects and give structures the cell. It protects the cells. Understand? It give structures to the cell. Understand? Now it is, that cell wall is found in plant, and in the higher plants, the cell wall is composed of cellulose, pectose, pectin, some parts of protein, etc. So mainly we say that it is made up of cellulose. It is a polysaccharides, that is a carbohydrates. In the algae, it is composed of, in the algae it is made of cellulose, galactans, calcium carbonate, and also manganese, etc. So in the plants, it is made of cellulose. In the made of cellulose, hemicellulose, pectin, protein, etc. While in the algae it is made of cellulose, galactans, calcium carbonate, manganese, etc. So these are the constituents. These are the constituents by which the cell wall was formed. In that, cell wall, there is a middle lamella is found. The middle lamella is found, which connects the other cells. Understand? And in between these, there is an intercellular connection. Between this, the intercellular connection for the interconnection of cytoplasm. These are called these plasmodesmata, which interconnects the cytoplasm of two cells. So, but there is interconnection taken place, and that the cytoplasm are connected to each other by means of plasmodesmata. So, because it is a non-cellular and somewhat hard structure, and so it protects the cells, it gives the structures to the cells, and also helpful in the interconnections of the between the cells. Now, there are so many structures which are found in the cytoplasm, that is known as cytoplasmic organelles or cytoplasmic inclusions. In that cytoplasmic inclusions, we have to study, and you know that endoplasmic reticulums, ribosome, Golgi bodies, there are certain organelles which are found in that. Now, in that, we shall first study endomembrane system. Means there are some organelles which are functionally related. Means functionally related organelles. They are the functionally related organelles. These are endoplasmic reticulum, Golgi Apparatus, lysosome, vacuoles. These are the structures which are functionally related to each other. So they come we study these organelles, these systems under endomembrane system. Now, first we shall discuss the endoplasmic reticulum. Now, the first we shall study endoplasmic reticulum. This is first discovered by Claude Phelan. They are the scientists who this one discovered about endoplasmic reticulum. But actually, we detailed knowledge came after the discovery of the electron microscope. In the through the electron microscope, when we observed the nucleic acid, we get that a net-like structure is found in the cytoplasm around the nucleus. Understands that in such a way. So this is the nucleus, double membranous structure. This is the nucleus, and around the nucleus we get such structures, that is of endoplasmic reticulums. In these structures are found. Understand? So if we separated it, if we out it, we shall get in this way it is found. Understand? And this is nucleus. So in such type, the net-like structure is found all over all in the cytoplasm. It is of two types. One is rough endoplasmic reticulum. Second one, a smooth endoplasmic reticulum. If this endoplasmic reticulum is associated with ribosomes, then it will called rough endoplasmic reticulum, and due to presence of ribosome, the protein synthesis takes place. We know that ribosome is the site of protein synthesis, and the smooth endoplasmic reticulum means there is no particles on that. Means here is particle ribosomes, but there is no particles, so that's why it is called a smooth endoplasmic reticulum, and it is helpful in the formation of lipid, steroidal hormones, etc. So we see that it is a net-like structures found in the cytoplasm. Understand? And it is of two types. One with particles known as ribosome, so it is helpful in the protein synthesis. The next one having no particles, so they are called a smooth endoplasmic reticulum, and that's why, and that is also take part in the lipid. Now the next includes is Golgi Apparatus, and it was first seen by Camillo Golgi in 1898. Understands? And it is in some plant, in the plant it is also known as dictyosome. It is also called lipochondria, and it is found near the endoplasmic reticulum. It has structures like that. This is the cisternae. It is composed of three parts. One is cisternae, that is flat ribbon-like organelles, that is cisternae. This one, whose ends are swollen. Ends are swollen on maturity, and sometimes next part is microvesicles. They are small pods, small vesicles. These are and they help in the secretion, and finally the Golgi vacuoles are also present. So these are the, it is also called Golgi vacuoles are found. So these are the structures which are found in the Golgi apparatus or Golgi bodies. Understand? Next one, it is found near the endoplasmic reticulum and helpful in the packaging of material and send them to their targets. Means they are helping the packaging of protein, packaging of other materials, and send them to the specific point. Second, it is helpful in the secretion. It is also helpful in the formation of lysosome. So these are the some functions of that, and about that you have already studied in your classes, like in class 9th. Understand? So study days. So say that Golgi bodies, that is discovered by Camillo Golgi, and it is helpful in the secretion and packaging. Main function is secretion and the packaging of materials and send them to their spot.